Okay, my clock says 12:01, so I'll go ahead and get going. Yeah, my pleasure to introduce James Frakes for his master's defense. Woohoo. Just a quick trajectory of James. I met him in 2018 when he took my entomology class then. And it became like readily apparent, maybe even on day one, that he was a huge enthusiast for aquatic insects, which also reflects his love of fly fishing. So if any of you know James, even in passing, you will know that he's an enthusiastic fly fisherman. And he joined my lab as an undergrad and did a number of different projects, and then ended up spearheading a project of his own to look at the sort of joint effects of a number of abiotic characteristics of streams on the thermal performance of stoneflies, which is one of his primary loves. And he just did a really fantastic job. This is one of those sort of dream undergraduate projects that produces a really great and interesting data set, which we worked on together for a while, and he ended up publishing last year as a really nice paper in Biology Letters. So when it got time for James to move on, I was sad, but then I was very happy because he asked whether he could join my lab as a master's student, and to me that was just a no-brainer thing. I was really, really happy to have him, and so he's been in my lab for a couple of years as a master's student, still working on stoneflies and a different aspect of stoneflies that he'll talk about today, focusing on temperature metal interactions in the sort of ecological context of the Clark Fork, the Upper Clark Fork Rivershed and Rock Creek. And I'll just say that it's been a real joy to have James in the lab. He's just a super tenacious scientist who rolls easily with experimental setbacks, which happen to everybody. No. Roman, can you mute, please? Thanks. And he's just done a really great job on this project. I love that he's a team player. He's very highly collaborative, and he's helped out other people in the lab just continuously throughout his time here. It's been really great, and I'll be sorry to see you go, James. So it's all you.
Well, thank you so much, Art. Let me share my screen here.
That Word document with notes from last week, and then sort of those ideas of both.
So let me just make sure that this is working. Hopefully everybody now can see my slides. Let's just double check, make sure everybody's muted, just so it's not too confusing. Today I'm really super excited to present to you all what I've really been up to over the last couple of years. I'm super excited to present my work on my favorite insect of them all. This is the giant salmonfly. Which is really, I think, one of the coolest insects of them all. And I'll tell you more about that in a little bit. Just for a quick roadmap for how this talk is going to go. The first half is going to be on the thesis as I've written it. And then the second half, I'm going to tell you about a project that I've been working on more recently that is clearly motivated by this thesis project. So to kick off the talk, I thought it would be a good idea to maybe remind you guys of a problem that's probably not at the front of your mind right now, as much as some of the other problems facing our planet. And that's the problem of heavy metal pollution in our freshwater habitats. I found this to be fairly surprising that heavy metal pollution is actually increasing in severity in most of our freshwater habitats all around the globe. And evidence for that is shown here in a figure from the Journal of Ecology, the Journal of Global Ecology and Conservation in 2020 by Joe et al. And they show that individual heavy metals are actually increasing in concentration over time. So these can have some really serious side effects for aquatic insects. So heavy metals can directly harm these aquatic insects because they generate free radicals and they produce oxidative stress, which can reduce the organism's performance, and it can even lead to their death. But many of the researchers who look at aquatic insects, they think of them as vectors for heavy metal transmission. And that's because these aquatic insects are located so low down in the food web that when they bioaccumulate heavy metals in their bodies, they can transfer that up into higher trophic levels and then even into top predators, potentially into humans if they were ever to consume fish from polluted habitats. So you might be wondering, why should we study aquatic insects? Like Art mentioned, I obviously had a interest in aquatic insects because of my love of fly fishing. I thought, man, if I can study these aquatic insects, I might have a edge on the river against some of those other anglers. But once I got involved in the Woods Lab, I was just blown away by the insane biodiversity in this group. They make up 70 to 90% of the biodiversity in freshwater habitats. So to put that into perspective, if you were to lift up a rock in a healthy stream, for instance, you could find all of these taxa living right next to each other under that same rock, which is just amazing. They're also the primary consumers and dominant predators in those habitats. They support higher trophic levels, like I mentioned before, and they're also commonly used as bioindicators of stream health. So hopefully I've convinced you that aquatic insects are really important and cool, and that heavy metals are a dangerous fate for them. But there's another issue that's facing our planet that I'm sure a lot of us have much closer to the front of our minds, and that's this issue of climate change. And we know that this is a serious issue. The way that this is playing out in Montana's rivers is that we're seeing hot summertime air temperatures overlapping with periods of low flows. That's because the consequence of climate change in Montana is increased drought severity in the summer times. So this leads to an equation for periods of really extreme warming during our summers, and that can have some seriously profound effects on aquatic taxa. So it can both have sublethal effects and lethal effects on aquatic taxa. So here's a really nice meta-analysis example from Conti et al. In 2014, and they show that stoneflies, the group of insects that I study, they're actually more vulnerable based off of a 23 trait analysis to climate change associated changes. And then a more direct effect here's a really nice example of a more direct effect of climate change by Till et al. In 2019, where they show that native fish are experiencing too warm of waters in their native waters, and they're having these fish die-off events. So here on the left, you can see in red is historical fish die-off events. And then on the right, there's two predictive models based off of climate change scenarios. And you can see that the probability of experiencing a fish die-off event increases, especially at these low latitudes. So I hope I've convinced you that heavy metals are very dangerous. And I'm sure we all know that climate change and increased warming is very dangerous, but very little is known about how these two factors interact to affect organisms. And it's really important to study things like this because many of our aquatic taxa are going to have to experience both of these stressors simultaneously. So I've isolated a couple of dangerous ways that these two factors may interact. First, temperature can increase heavy metal exposure. And that's because all small ectotherms, like the insects that I study, their biological rates are tied very closely to temperature. So in warmer waters, you have increased feeding, growth, ventilation, respiration, and all of those things can further expose them to heavy metals. Another way that they may interact is that certain heavy metals may decrease the upper thermal limits of aquatic taxa. Now, they do so by interfering with the physiological processes that control these upper thermal limits, which I suggest are really important traits to maintain in the face of climate change. So to look for these interactions, I've used my favorite insect of them all, the giant salmonfly nymph. So indulge me for a minute because I'm going to make a claim that these salmon flies are one of the most important insects to Montana. And that's because they provide an unparalleled fly fishing opportunity. So stick with me. It's because these insects are the world's largest stonefly species, and they're one of the largest aquatic insects in our rivers around here. And they also have a very synchronized hatch. They come off in June, around the second week in June, and they hatch for about a 2-week window. And during that time, the fish are seeing huge amounts of these large-bodied insects landing on the surface, and the fish just go stupid for them. And the fishermen will come from all over the world to experience this hatch, bringing with them money to these small towns like Clinton or Phillipsburg or Wisdom. So to put a dollar value on that, the fishing industry in Montana brings about $375 million per year, and that's comparable to the revenues of all the breweries in the state per year. They're also super ecologically important. They provide obviously a large food source for these native fish, and they also shred leaves and cycle carbon. So I've used 2 different populations of these salmon flies in my study. My first population comes from Rock Creek, which you can think of as salmon fly heaven. It's clean, it's cold, and it's got a really healthy, abundant population of these salmon flies. And I compared that population to a population in the Upper Clark Fork River, which, as most of you know, is a river that's experienced quite a significant amount of heavy metal pollution. And that's because in Butte, Montana, not shown on this graph, it's located about here, there's 2 of the world's largest copper and silver mines. And those mines have released heavy metal tailings into the headwaters of the Upper Clark Fork River. And those metals have been washed downstream over time. And since that time, those metals have impacted the entire community in the Upper Clark Fork River, all the way from the macrophytes up to ospreys. And as of right now, this stretch of the Upper Clark Fork River is actually America's largest complex of Superfund sites. So let's take a look at my sampling sites. You can see that the rivers have really different characteristics. Here, the Upper Clark Fork River, you can see, is a slow meandering river. It also has a lot of agricultural withdrawals of water. And then there's also significantly less riparian shading on the Upper Clark Fork River. And all of these factors together make it so that in the summertime, this river, the Upper Clark Fork River, actually heats up significantly more than Rock Creek ever does. And it's for that reason it's for that reason that I study both temperature and heavy metals in the Upper Clark Fork River. Okay, so to look for these interactions between temperature and heavy metal, I've performed a lab experiment where I've exposed them, basically 2 lab experiments, where first I exposed them to copper, which is a biologically essential element, but it can become toxic at high levels. And then I repeated the experiment using lead, which is shown to be toxic across all levels. But keep your eyes peeled. You'll notice that the salmon flies respond very differently to copper as they do to lead. So I've used a factorial design. And so I've used 12 insects from each of my rivers, and I've held them at 2 different temperatures and at 3 different metal levels. These metal levels are a control. This mid-level here is actually the 95th percentile of the concentration of the given toxin in the Upper Clark Fork River. And those values come from the USGS website. And then I also expose them to an experimentally useful high level here. So to achieve these treatments, I've used 2 temperature controllable incubators. Within those incubators are these tubs, and I've fitted each tub with an aquarium pump and an air stone to maintain flow and oxygenation. I've also housed my salmon flies individually in these mesh bags. And inside those bags, I've placed 3 leaves, which I've collected from Rock Creek. Then I let these treatments run for about 3 weeks. And then I came back and I measured 3 response variables: survival, growth, and then upper thermal limits. So I will show you those results now. My first result is survival. As you can see here, I plotted survival on the y-axis, and across the x-axis, we have those different treatment levels. What you can see here is that as you expose salmon flies to copper, they tend to die off. However, if you expose them to the levels of lead that I did, they did not die off at a significant rate here. Another interesting thing jumps out, and that's that salmon flies from the upper Clark Fork River actually tended to die off at higher rates than those salmon flies from Rock Creek. Okay. My second response is growth rate. So bear with me, these figures are a little bit confusing, so I'll try and explain them as best I can. The size of these points corresponds to the initial mass of each of these insects. And then on the y-axis, I have my growth rate. And on the x-axis, I have my copper concentrations. So the way that I was able to get these copper concentrations was by using a tool called an ICP-MS, which stands for an inductively coupled plasma mass spectrometer. And this is an amazing tool that allows you to get individual samples of a particular element. So you can think of these as the body burden of copper for individual salmon flies. Okay, so now that we know what this means, we can look at the overall trend, and that's that salmon flies that accumulate high levels of copper tend to grow less. There's also an interesting interaction that occurred here, and that's that Rock Creek insects disproportionately show reduced growth at high copper concentrations compared to those from the upper Clark Fork River. So now I'll show you my corresponding figure for my lead experiments. And the first thing that you'll notice is that there's a split on the x-axis here. Now, the reason for that is because the ICP-MS is not sensitive enough to be able to pick up the levels from my control insects, the levels of lead from my control insects. So I've just plotted those here so you can make a visual comparison between the treated and the control groups. And when you do that, you can see that there's really a very little effect of lead on their growth overall. However, there's this really kind of interesting thing that does pop out, and that's this group of salmon flies from the upper Clark Fork River that are growing particularly well in low to moderate levels of lead. Now, this might be coming about by a couple of ways. It could be evidence of some sort of acclimated or adapted response to lead, or it could be due to the fact that when I spike these treatments, I use lead acetate. And when you dissolve lead acetate, the acetate disassociates from the lead, and it can actually potentially act as a nutrient in those tubs, and it could promote the growth of biofilms, which potentially boosted the food supply to those salmon flies. Okay, moving right along, we're going to look at my final response variable, and that was upper thermal limit, or CT max. CT max is a measure of an animal's upper thermal limit, and you get these values by performing these temperature ramping experiments. So in the case of salmon flies, what we do is we ramp up the temperature until they display a response that's indicative of extreme thermal stress. And for salmon flies, we use the response loss of righting response. And essentially what that means is when you take a salmon fly and you flip it on its back, when it no longer can flip itself back onto its feet, we call that their CT max, and those temperatures are what I've plotted here on the y-axis. Again, on the x-axis, I have that body burden of copper. And what you can see is that copper significantly reduces their CT max. Those that acquire high amounts of copper actually have about a 10-degree decrease in their CT max, which is hopefully quite frightening, or it reminds you that this is quite a frightening interaction, because this is what I suggest could be a really quite a dangerous interaction between a heavy metal and the upper thermal limits. Okay, I'll now show you my corresponding figure for my lead experiment. And first thing you'll notice is that there's a completely different pattern going on here. You notice that all of these blue points are underneath all of these red points. So these blue points were cold acclimated and these red points were warm acclimated. And what you can see is that salmonflies are sensing their thermal environment and adjusting their CT max. And there's essentially no significant effect of lead on their CT max. So when I got this result, I was a bit perplexed, right? In one case, I have metals significantly reducing CT max. In the other case, I have temperature significantly interfering with their CT max. So once you go down the wormhole of trying to get at all of these interactions, you quickly realize that you can't account for everything. So what I've decided to do is perform a next set of experiments to get at this question of what is really the most important in setting these CT maxes of the salmon flies in the actual Upper Clark Fork River. Is it the stress from heavy metals, or is it the warmer conditions that they experience that affects their thermal limits there? So to get at this question, I performed an in-river mesocosm experiment. So to do this, I collected a bunch of salmon flies from Rock Creek, and then I placed them in these cages that I built. And I put those cages in 3 locations across the Upper Clark Fork River, and then in 1 location back in Rock Creek as a control. At each of these locations, I filled the cages with substrate and leaves from the local environment, and then I capped the cages with wire mesh, and I left them there for 4 weeks across the hottest part of the summer. Then after that time, I came back and I measured those upper thermal limits. So what we can see here is that salmon flies who were held in Rock Creek, this is the control group, actually have significantly lower CT maxes than those salmon flies that were held in the Upper Clark Fork River. And I suggest that this is because the Upper Clark Fork River is significantly warmer over the summertime when they were in those rivers. So it means that in the short time, they're relying more on this acclimation response, adjusting their CT maxes upward to mirror the thermal environment that they've been experiencing. Okay, so shifting gears slightly, now I'd like to tell you a bit about this second set of experiments that I've been working on lately. And it goes back to this dangerous interaction that I found, right? I found that copper significantly reduces the CT max of salmon flies. And it wasn't just enough for me to say that there's this dangerous interaction. It was important for me to try and figure out what was the underlying mechanism. So to explain the hypothesis that I have for why this might be occurring, we first need to understand what goes on at these upper thermal limits, which are these really important traits that animals will have to maintain as we navigate through climate change. And one of the leading hypotheses for what sets these upper thermal limits in aquatic ectotherm taxa actually is oxygen, is one of the main drivers of these upper thermal limits. And this hypothesis is referred to by many different names. Some call it the OCLTT hypothesis, or oxygen capacity limited thermal tolerance hypothesis. Some call it the OSI, or oxygen supply index. But in the Woods lab, we simply refer to this as the oxygen problem. And the problem is this. It's that ectotherms' metabolisms are exponentially tied to temperature. So you can think of this metabolism as the oxygen demand of the organism. Now, the available oxygen in a given environment is only, in an aquatic environment, is only increased ever so slightly by temperature. And what this means is at a certain temperature, there's a mismatch that occurs in the oxygen demand to the oxygen supply. And it's at this temperature that many people think the upper thermal limits are reached. So when I was an undergrad in the Woods lab, I decided to test this hypothesis using these salmon flies. And this is really how I first got involved with Woods lab. And I found that oxygen is really strongly influenced, CT max is really strongly influenced by oxygen. As you can see here, if you hold salmon flies in near 100% saturation of oxygen, they have CT maxes around 30 degrees, but if you hold them in hypoxia, their CT max is greatly reduced. And I also find that flowing water is a really important way that oxygen is delivered to these salmon flies. And that's because water flow can deliver more oxygen in a given amount of hypoxia. Okay, so let's look at the gills of these salmon flies, where they acquire their oxygen. So these gill tufts provide an increased surface area, which allows for oxygen to diffuse from the water into the insect. And then inside these gills are these branching networks of tracheal tubes that deliver oxygen all over the insect's body. You can see that here in this figure. These gills are also made up of epithelial tissue. And so many researchers think that these respiratory surfaces are a primary way in which heavy metals and other pollutants may enter the body. So if we put all of this together, what I think might be occurring is that copper is binding to these gill surfaces, and it's either damaging them or somehow inhibiting their ability to extract oxygen. And that's why we're seeing this reduction in their upper thermal maximums. So to test for this hypothesis, I've come up with a fairly simple experimental design. It's essentially two different experiments. In one experiment, I expose salmon flies to 3 parts per million of copper, and I compare that to a control. And then in my second experiment, I use 5 parts per million of copper, and I compare that to a control. I let salmon flies experience these conditions for a week, and then after that time, half the salmon flies went to one experiment and the other half went to another. So the first set of experiments I did was CT max, and basically just to make sure that the mechanism was still occurring in the same way as I saw in my first study. And as you can see, it is. Salmon flies who were never in contact with copper have significantly higher CT maxes than those salmon flies who were exposed to copper. Here is my CT max setup. You can see right here is the temperature controller, and then right here I have individual mesh chambers where I put the salmon flies in and observe them. Then the second half of my salmon flies went up to the Flathead Lake Biostation, where I performed some respirometry. I shouldn't say I performed it. It was largely a collaborative effort with Rachel Mallison. Thank you so much. And up there, we did a kind of a simple method of respirometry. We used something called closed chamber respirometry. Essentially what that is we put our insects in these glass vials, and then we capped those vials and measured the amount of oxygen that they draw down over time. And by doing this, we're able to calculate their mass-specific metabolic rates. And that's what I'll show you here. You can see here on the y-axis, I have mass-specific metabolic rate, and on the x-axis, I have different dissolved oxygen levels. And as you can see, there's quite a significant amount of overlap between my copper exposed and my control groups for both my 3 parts per million comparison and my 5 parts per million comparison. But we did notice when we were analyzing this that if you just take the values below around 4 parts per million, or sorry, 4 milligrams per liter of dissolved oxygen, there is some significant differences. So we went back to the drawing board, and what we did is we went back to the raw data. And so what we've done here is we've taken a single data trace from a single vial. And so you can think of this as the dissolved oxygen within a singular vial on the y-axis. And then on the x-axis, we have the duration of the experiment. As you can see at the beginning of the experiment, there's high oxygen. Those insects draw that down fairly linearly. And then there's some critical point right here. And using piecewise regression, we're able to calculate that breakpoint. And we're calling that the critical oxygen point, or you can think of that also as kind of like the hypoxia tolerance limit of these insects. And that's what we use as our response variable for this next round of analyses. So here are some confusing figures that show just that. I've shown all of those data traces on this larger graph, but in the upper right-hand corner, you can see those critical oxygen points plotted as these box plots. And the significant effect is happening right here, where salmon flies who are exposed to copper actually cannot withstand as deep of hypoxia as those that were not in contact with that copper. So something about this copper exposure is causing them to have some sort of differential sensitivity to hypoxia. So if we remember back to my original hypothesis, I really thought I would see lower metabolic rates across the board in the copper exposed insects. And that's because I thought they may have some sort of damaged gills. So as part of this experiment, I decided to look much more closely at those gills. And I'll show you some images now. So I used fluorescent microscopy techniques to look at these gills. And I've dissected gill tufts off of my salmon flies and exposed them to this copper binding fluorescent dye. So places where you can see green are areas that have higher copper concentrations. So let me walk through this. On the left here, we have a gill filament. And as you can see, there's essentially no copper on these gills here. But if you look at the base of the gills where they attach to the cuticle, you see these green cells here. And these are the chloride cells. These chloride cells are in charge of ion regulation for these salmon flies. So it might not be that surprising that they have significant amounts of copper ions bound to them. So let's take a look at the insects that were never in contact with copper. And you can see here, again, the gills have essentially no copper on them. But if you look very closely, you can see low levels of copper on their chloride cells. And this might not be that surprising because copper is a biologically essential element. So they likely have been in contact with low levels of copper across their lifetime. Okay, so this suggests that copper is binding to these chloride cells in some way, and it's potentially interfering with these salmon flies' ability to respire down in that deep hypoxia zone, and also it might be influencing their upper thermal limits in some way. So that's about as far as I got on this experiment so far, but let me just go back to the top and do a big conclusion here. So from the top, the first thing I found is that copper directly reduces all three of my performance metrics: survival, growth, and CT max. But these salmon flies appear to be super hardy when it comes to lead. I also found a kind of dangerous result, and that's that copper depresses the CT max, which may make these populations in copper polluted habitats more vulnerable to heat waves from climate change. And that's because they may reach their thermal limits sooner than those from non-polluted rivers. I also note that salmon flies rely quite heavily on phenotypic plasticity in setting their thermal tolerance traits. And this is likely an important way that aquatic insects will be able to navigate climate change, I think. And then in my second round of experiments, I found that copper is not binding to the gills in the way that I thought it would, but instead, copper is binding to those chloride cells, which may make these salmon flies more sensitive to high temperatures and hypoxia. Okay, with that, I have to say a huge thank you to everyone involved. First and foremost, huge thank you to Art Woods. You've been such an amazing mentor. You really got me super excited about science. It's just going to stick with me forever, and thank you. A big thank you also to Amanda and Rachel and Ben and his lab. Couldn't have done any of this stuff without you guys. A shout out to my field crew. I know some of you are here. Thanks for putting in all those long hours. And also a big thank you to my partner, Isabel. Thank you for, going through this talk with me so many times. And also wanted to thank my fantastic lab mate, Jackson. You've been great just to bounce ideas off of over the years, and thanks for letting me bug you all this time. This project was funded by the Montana EPSCoR program and specifically the Cruise project. Okay, I would love to take some questions if you have them.
Thanks, James. And yeah, plenty of time for questions. So go ahead and speak up.
I have a question.
Yeah.
Before you plotted response against growth rate of the stoneflies, but you did it against absolute growth rate. And then because big stoneflies are going to grow faster than small stoneflies, you had to control for the different size of the stoneflies. So why not then plot a mass specific growth rate? So where you calculate a mass specific growth rate, so a per day growth rate, which would be the log of the growth now minus the log of the growth then divided by the amount of time, and that might clean that up a lot, and then allow you to have growth be a little bit more scale free across body size.
Sure. Yeah. If I'm understanding you, I actually didn't control for their body size in that growth rate.
Okay.
I just did growth rate, basically initial minus final, and I didn't.
Yeah, I would do the log of the final minus log of initial divided by the time, and that'll give you a per unit time growth rate. And I would do that.
Okay.
That's how we do it for all this, like production analyses and things like that. So.
Okay, fantastic. I see Kriya has a.
Yeah, awesome talk. Really nice job. I noticed that you're comparing the two populations in growth versus survival. You had this cool thing where I think it was the Rock Creek had a greater effect of copper on survival, and the Upper Clark Fork had a greater effect of copper on growth, which, hits me as a growth survival trade-off. And I'm wondering if you have any thoughts about what that might mean or why it showed up that way.
Yeah, that's really interesting. I hadn't thought about growth survival trade-offs, but it is weird to see a different response from one river compared to another. I don't know. I'm a little bit skeptical about the growth rate response in the lead experiments, especially. Salmonflies have these really confusing and intricate sort of life history changes in their growth rates. So certain times of the year, they'll grow a lot, and then they also go essentially dormant over some of the summers right before they hatch. So it's confusing to be able to compare these two rivers because they likely have different seasonalities between Rock Creek and the Upper Clark Fork River. So comparing growth rates can be somewhat confused, but I'd love to think more about this survival growth trade-off thing. That's a great idea.
Nice job.
Jackson's got a question?
Yeah. Awesome job, James. You totally killed it. It was great to see all this come together in your talk. I was just thinking, we both know that the further you go up the Clark Fork River, the less salmon flies there are, at least in the mid and upper Clark. Like there's salmon flies downtown, even here in Missoula. You go up to like Deer Lodge and they're really hard to find, as you know. Do you think these temperature oxygen interactions could explain that with the sensitivity at the concentrations of metals that you saw, et cetera? Do you think that this could explain some of their range limits and how density changes across the elevation gradient? Or do you think this is like more of a subtle effect than like the big hammer that's affecting their densities and distributions in nature?
Sure. I don't know if it's probably a complicated answer, right? Like it's probably not one thing. What we do know is that the concentrations of metals increases as you go up slope. And there could be some sort of interactions between all of those metals that I haven't accounted for. And it could also be that they accumulate that over their lifetime, which increases the severity of that. But I also think that it could be simply a habitat issue up there. There's a lot of fine sediment, and there's not those big boulders that we find those salmon flies on. So I don't think that I can make the claim that it's entirely metal or metal temperature driven.
Ken, well, you gave me an awesome idea for a follow-up study that we should think of. I'll tell you about it later.
I love it. Okay, cool. Ray Calloway?
Yeah, hey, that was really nice work. Nice presentation. I just have a question about experimentation. When you exposed your creatures to the metals, was it both in over time? Was it both in diet and in water, or was it just in solution? Because I would think that since algae and even organic matter might bioaccumulate or biomagnify, when it's in their diet, they might get a lot heavier doses. And just curious if that was integrated in some way in your treatments.
For sure, yeah. What we did was we spiked the water at the beginning of the experiment, and then we added the salmon flies and the leaves. And we actually measured that water using the ICP-MS. And what we found is that at the beginning of the experiment, there's high levels of these metals in the water, dissolved in the water. And then over time, those metals went away. And what happened is they likely bound to the leaves and the other solids in the tubs. And then for the second half of the experiment, those salmon flies were then exposed to it through their diet. So I got both, through in the water column and in their diet.
Excellent. Sorry, I missed that. That's great. Okay.
Yeah, for sure. That's good clarification. Anybody else have any questions?
Oh, I have a question, James.
Bria?
First of all, congratulations. That was a really good talk. It was really nice to see everything come together. And this is more theoretical. Did you ever think about kind of the effects of salmon flies versus a dragonfly nymph? Because I'm just wondering, maybe the surface area of their gills might attach more to copper than other insects, but I'm not sure if you've thought about that.
No, I hadn't thought about that. As maybe dragonflies have these really cool gill systems where they pump the water through. So I have really no idea about how different, which insect has more surface area. And I don't know. I have no idea. Sorry, I don't have an answer for you. But good question. Okay, Zach Chevron.
Yeah, awesome talk. Second thing, what everyone said. I have a question about sort of the your images of gill structure and sort of the accumulation of copper within the gills. And so you found accumulation sort of in the chloride cells, which you mentioned is important in sort of ion regulation. Do you think that there's any kind of mechanistic connection between ion imbalance and CT max? I mean, I understand why you'd focus on oxygen, right? But given that there does seem to be that's where the copper is accumulating, do you think any of this sort of reduction in thermal tolerance is related to ion imbalance?
Yeah, the only thing that really jumps to my mind first is that maybe there's some sort of cost associated with having high levels of copper in those chloride cells. Maybe they're trying to detoxify those cells, and that's increasing their metabolic rates in some way, and that's affecting them at those low oxygen levels. I don't know. Did you have an idea for a more direct?
No, I was just curious what your thinking was there and just sort of the interaction between those kinds of stressors. But yeah, I'll think about it.
I think maybe some sort of metabolic cost of or damage from that copper in those chloride cells.
Sure. Cool.
Amanda?
Hey, can you hear me?
Yeah.
Great job. I was just wondering what those images, the last ones that you showed with the copper binding fluorescent probes, were they confocal images?
Yes. Yep.
Were you able to see if it was just binding to the surface or if it was somehow like within the subsurface?
Yeah. So no, what we found in the chloride cells is it seems to be binding to the outside of those chloride cells. Like, it almost looks like it's in the cell membrane area of those chloride cells. And I got that just by doing these Z stacks. So went from the bottom of the focal plane up to the top. And when you do that, you can see that there's significantly less on the inside. As for the gills, they don't seem to be binding to the outside or the inside of those tracheal tubes. They're not really in there anywhere.
Yeah, great. Great job.
Thank you.
Looks good.
Thanks.
Okay, well, if there are no more questions, let's thank James again. And James and committee will convene at 2 o'clock for the closed door session.
Sounds
Okay,
good.
thanks everyone.